Hello children and welcome to my channel Chemistry by Miss Nidhi Banga. In today's video, I'll be talking about part 2 of biomolecules. That means we'll be discussing proteins in this video. So let's get started. Proteins are the polymers of alpha amino acids. Alpha amino acid means amino acids because it has amine group as well and acid group as well. And alpha amino acid is because the relative position of amino group is at the alpha carbon. That means this is the carboxylic acid group. The amino group is attached to the alpha carbon to it. If it had been something like this, then this would have been beta amino acid. But we have amino group at the alpha carbon. So this is alpha amino acids. And they are the ones which polymerize to form proteins. So proteins have alpha amino acids only. On hydrolysis, they give alpha amino acids only, right? Now, if you look at the general structure of amino acids, it has 4 different groups. So it's a chiral carbon. This carbon is chiral carbon, which makes it optically active. But if this R group, instead of an alkyl group, if you have here only H, then this carbon is no longer chiral carbon. So in that case, it won't be optically active. So when R group is hydrogen, that is in glycine, glycine is an amino acid where this R is equal to H in glycine. So in glycine is the only amino acid, only alpha amino acid, which is optically inactive. Rest every other amino acid, every other naturally existing alpha amino acid is optically active. Okay. These all amino acids, the naturally occurring amino acids, they belong to L series. That means the NH2 group here is onto the left side. Now, let's talk about how they are classified. Amino acids, as the name suggests, has amino group and acid groups. But sometimes there are more amino groups than acid groups. Other times there are more acid groups than amino groups. So on basis of that, we have divided them in 3 categories: acidic amino acids, basic amino acids, and neutral amino acids. Acidic amino acids, as you can guess, they have more number of COH group than amino group. That is why overall they are acidic. In basic, there are more number of NH2 group than COH group. In neutral, the number of NH2 group and COH group are equal. Examples, very easy to remember. Glutamic acid and aspartic acid are acidic amino acid. Glutamine and asparagine are the amino acids which are basic amino acids. So glutamic has a basic counterpart, glutamine. Aspartic acid has a basic counterpart, asparagine. And similar to that, we have glycine and alanine, which are neutral. Okay, so on basis of number of COH group and NH2 group, amino acids can be categorized into these 3 categories. Next categorization is on the basis whether these amino acids can be synthesized in our body or not. The ones which can be synthesized in our body, that means the ones which we do not need to take in our diet. That means it's not essential that we obtain them from our diet. Those are termed as non-essential. Glycine and alanine you can write as 2 examples of non-essential. Then we have essential. These are the ones which our body cannot synthesize. That means it's essential that we obtain them from diet, through diet. Leucine, isoleucine, you can remember, are the essential amino acids. Talking about the general properties of amino acids, they are colorless, crystalline solids. They are water soluble, they have high melting point, and they behave more like salts than amines or acids. Now why is that? It's because since amino acids have acidic group as well and basic group as well, what happens is these lone pairs abstract this proton and these 2 electrons shift to oxygen here, and it gives us this thing. Since nitrogen has accepted a proton without its electrons, basically it has used its own electrons to form bond with the proton, it gains a positive charge, because the two electrons which were whole nitrogen's property, now they are being shared between nitrogen and hydrogen. Okay, so nitrogen becomes a little deficient in electrons. Now, since these two electrons, which were shared between oxygen and hydrogen, now completely get transferred onto oxygen, oxygen gains here a negative charge. Okay, so this thing that you get, it has a positive charge also and a negative charge also. It's called as zwitterion. Now, it's different from the regular ionic compound that you have like NaCl. Okay, it's different from that because in NaCl, a cation is immediately followed by an anion. Whereas here, the cationic, the positive part and negative part are separated through covalent bonds. That is why it's different from the regular ionic compound. Nevertheless, it still has charges, and these charges are responsible for the strong electrostatic attraction that it feels, which is why it has high melting point, because you'll have to give more energy to break this strong electrostatic attraction and therefore separate the amino acids. Solubility also can be because of this zwitterion structure. Water is a polar molecule. Here we have two charges. That means like dissolves like. This makes this amino acid water soluble. They behave like salts because they have positive center and negative center. That is why they behave like salts. They also show amphoteric behavior. Amphoteric behavior means that it can react, these zwitterions or amino acids can react with acid and base both. They react with acids and bases both. Of course, they react with acids and bases both. The basic part would react with acid and the acidic part of amino acid would react with base. So an amino acid can react with acid and base both, and therefore it shows amphoteric behavior. Now let's talk about structure of proteins. Basically, structure of peptides. So what happens is, if you have an amino acid like this, where this is the chiral carbon, this is the acid part, this is amine part, and in R, instead of R, you have hydrogen. This is glycine. Whereas there is this amino acid which has acidic part, basic part, chiral carbon, hydrogen, and this is the R group. This is not chiral. Okay, this is chiral. Anyway, so what happens is, when these two come together, the carboxylic part combines with the amine part, and with the loss of water molecule, it forms a dipeptide. The linkage between this carbonyl carbon and this NH is called as peptide linkage. It's a kind of amide linkage. Okay, amide linkage you've seen in amines chapter. So this is peptide linkage, kind of amide linkage. Now, how would we name this dipeptide? If glycine, the amino acid, which is donating 2 atoms to the water molecule, will be given priority. It would be named first. Okay, then the amino acid which is donating only 1 hydrogen. Okay, so here we would name this dipeptide as glycylamine, because the glycine is donating 2 atoms. This is how you remember. Okay, this is not the logic, but this is how you remember. Okay, so this is glycylalanine. Now let's see this dipeptide. Let's see if you can name it. When you have a dipeptide and you are trying to figure out what are the 2 amino acids, you can simply first form the respective amino acids. This one will be, now look, it has COH group, amine group, and the R group is CH3. So this is your alanine, which is donating OH. Okay, so it's donating 2 atoms. So it would be named first. Then this. Now which one is this? This has amine, acid, one H, and the other one is also H. So that means it's glycine. Okay, these two are very simple to remember. So you may not be given the structure of the amino acids, but in other cases, you will be given the names also. For example, here we have told you that this one is glycine, this one is alanine. So you'll just have to say that, okay, the one donating 2 atoms would be named first. So the dipeptide would be named as glycylaniline. Here, even though they have not given you the names are very easy to remember. When there's a methyl group in place of R, it's alanine. When there's hydrogen only in place of R, it's glycine. So now it would be named as alanyl glycine. Okay, because it's the alanine which is first in the dipeptide, or the one which is donating 2 atoms. Okay, now let's classify proteins. Proteins, on the basis of the shape, the molecular shape that they take up, they can be classified in 2 forms: fibrous proteins, globular proteins. It's also one of the most commonly asked questions. Differentiate between fibrous proteins and globular protein. Fibrous proteins, as the name suggests, the polypeptide chain are running parallel to each other. Okay, it looks like fibers. And these fibers, these polypeptide chain, they are stabilized by hydrogen bond or disulfide bond. They are insoluble in water. They are insoluble in water. And how can you remember this? You remember the examples. You remember that keratin, which looks like a fiber, which is also a protein in hair. Hair, you know, thankfully from keratin treatment. It's also a protein in wool and silk. All these 3, they look like fibers, right? So it's fibrous protein. Myosin, the protein found in muscles. Muscles are also muscle fibers. Muscles are also long cells, right? So the protein inside it would also be long and fibrous. So these 2 are examples of fibrous protein, and they are insoluble in water. Try dissolving silk or hair or wool in water, they will not dissolve. So you remember it like that. Okay. Next up, we have globular protein. As the name suggests, it's a globule kind of shape. That means when polypeptide chain sort of coil around themselves and make a spherical structure, take a spherical shape, then that is globular protein. It's soluble in water. How would you remember that? Remember the example. Globular is spherical shape. Albumin, the protein found in egg, in the white of egg. That egg is spherical, so you remember that the protein inside it is also a globular protein, a circular or spherical protein. Enzymes are all globular proteins. So I don't know about you, but whenever I think of enzymes, I think of a spherical shape. Okay, insulin is another globular protein. Now let's talk about different structures of protein. There are 4 structures that a protein can assume: primary, secondary, tertiary, and quaternary. Primary is very simple. It's simply the sequence of amino acids. Whether it's glycine, then alanine, and then aspartic acid, or aspartic and then alanine and then glycine. Okay, different proteins have specific sequence of amino acids, and that is what constitutes the primary structure of primary structure of protein. Then when the amino acids combine together through peptide linkage, they form a polypeptide chain. And that polypeptide chain will either coil up or will fold. Okay, and that would be its secondary structure. Secondary structure is assumed by either coiling or folding of polypeptide chain. So based on that, there are 2 secondary structures: alpha helix and beta pleated. Alpha helix is when polypeptide chain exists in a right-handed screw or helix. Whereas beta pleated is when the polypeptide chain is stretched to the maximum. And then here, there are many beta plated polypeptide chains where there is intermolecular hydrogen bonding. Intermolecular because this is one molecule, this is one polypeptide chain, and this is the other polypeptide chain. Okay, so there are two different molecules, and between these two different molecules, if hydrogen bonding is taking place, it would be termed as intermolecular hydrogen bonding. So in beta plated structure, intermolecular hydrogen bonding takes place, whereas in alpha helix, the hydrogen bonding takes place between the amino acids within the polypeptide chain. So if there is NH here and C double bond O here, the hydrogen bond that takes place between them is the intramolecular hydrogen bond because it takes place within the polypeptide chain. Okay, this is the main difference between alpha helix and beta plated. Many times this difference is asked in terms of MCQ. Okay, now another thing important is that alpha helix, beta plated, they are stabilized by not just hydrogen bond but also disulfide bonds, ionic interactions, and van der Waals forces. Now talking about the tertiary structure. Tertiary structure we have actually already discussed. We have discussed fibrous proteins and globular proteins. Those constitute the tertiary structure. Now, once we have either beta pleated or alpha helix, if they further fold, that gives us tertiary structure, which is either fibrous or globular. Okay. Next and final structure is quaternary. In quaternary, what happens is sometimes some proteins have more than one unit. They have two or more subunits. So how those two or more subunits are spatially arranged in 3 dimensions, that is what quaternary structure is all about. Okay, so these are your structures of protein. The tertiary structure also is stabilized by not just hydrogen bond and disulfide bond, but also by van der Waals forces, interionic interactions, and yeah, that's about it. Last topic of today's video, denaturation of protein. So when you have your normal protein in biological system with its unique 3D structure and its unique biological activity, that protein is called as native protein. This native protein, if is subjected to change in temperature or pH, that means if you heat it or if you decrease or increase its pH, because they have a range of pH and temperature in which their biological activity and structure is retained. Anything outside that range, the biological activity and structure of them changes, and that change is called as denaturation. So what happens in denaturation? In denaturation, the secondary and tertiary structure of protein are destroyed, but the primary structure is intact. It remains intact. It doesn't change. Okay, so this is what happens in denaturation, and this is very, very important, that during denaturation, the secondary and tertiary structure only destroy. The primary structure, that is the sequence of amino acid, will not change. Examples of denaturation: when you boil egg, then the egg coagulates, that is denaturation. And when the milk curdles, when you add something sour into it, the milk curdles, that is also an example of denaturation. So when you heat something, when you boil milk, basically you are providing temperature outside the comfort range of the native protein. So therefore, then denaturation will take place. And in curdling of milk, you are adding something sour and you are heating it. So you are basically denaturing the protein present in milk, which is casein. Okay. Now, what happens is, This is a question from your in-text question. Where does the water present in egg go after boiling the egg? So when we are boiling the egg, we are basically denaturing the protein present in egg. Which protein? Albumin. Albumin we know is a globular protein. Upon boiling, it gets denatured or coagulated. Now this coagulated protein absorbs all the water present in the egg. Okay, this is where the water is sort of used up. Okay, so this is where the water goes. Next up, we'll be talking about vitamins and nucleic acids.